standing for truth doesn't seem to understand the difference or want to understand the difference.
You and Spongebob need to start a college together.
He bastardizes the ENCODE project, is what he does.
Because the DNA sequences that we possess are largely from living species, we don't yet have a very good analogy for the transitional forms argument. Maybe you could argue with some of the limited data we have. So I'm not going to cover that. But the parallels, again, to the anatomical arguments find an analogy in DNA. Is there genetic evidence that rejects the hypothesis of design? If I'm using design as a competing explanation for the data we just looked at, what about at the genetic level? And of course, Biologos would cite this. If you look at their genetic evidence, they talk about genetic scars, which again, by its name, implies some sort of error. And I'm not trying to misrepresent them. You'll see some of the detailed arguments are arguments from function. So what is the function of DNA? How much of the human DNA is functional? Some evolutionists might say, well, maybe 10% is functional. That's all you can argue for. Let's think back to the evolutionary arguments for the appendix, for the tailbone or the coccyx, for the evolutionary vestige. What was the problem with those arguments? I'd say there were two problems with those arguments. Number one, there are arguments from silence. Absence of evidence is not necessarily evidence of absence. I'll say it again, absence of evidence is not necessarily evidence of absence. It might be, but it might not be. So it's already on a shaky foundation. What makes it even more shaky, and I think a second reason these arguments faced some severe challenges, is the absence of evidence was not derived from decades of experimentation that failed to identify positive evidence for function. The absence of evidence arose from the absence of experimentation itself. So then no surprise, as experiments progress, that evidence of absence disappears and we find evidence for function. So when evolutionists might say there's little evidence for function in the human genome, the first question to ask, of course, is, well, what experiments have been done? The gold standard is genetic knockout. Fortunately, that's still considered unethical in humans. We shouldn't be guinea pigs where someone goes into you and tries to knock out your DNA just for knowledge's sake. But there are individuals around the planet that have, naturally, because of mutations, because I would say because of the fall, they're missing sections of DNA. Or somehow some of their genes have been rendered non-functional. Now, only 6%, I think this is an Icelandic study, only 6% of the genes in the human genome have this sort of individual representation, which means the vast majority of our genes have yet to be tested by the gold standard for function. And since genes represent less than 5% of our total DNA, you can easily see then that the vast majority of our DNA sequence has yet to undergo any experimental manipulation. There's been biochemical studies, you may have heard of the ENCODE project, that they said there was evidence for function in 80% of the genome, and it was criticized as being premature. It is premature. It's only biochemistry. It's not genetics, and they're only looking at a small subset of cell types. There's all sorts of developmental windows they did not have access to, and again, you're running into ethical issues here. What are you going to do? How are you going to access a baby in the womb and test the function of DNA in those tissues? But I think we have the same problem as we do in anatomy and physiology. Absence of evidence is not necessarily evidence of absence, and the absence of evidence is from absence of experimentation. To me, what the ENCODE project suggests is a trajectory that is pointing towards increasing levels of function. We won't know if that trajectory holds up or not, but it looks to me like the evidence is pointing towards high levels of function in human DNA. And again, that's what it looks like to me. That's a hypothesis that's testable. And I anticipate then that as that level, the evidence for function goes up, the arguments then will have to necessarily change. And let me just pick one example real fast for sake of time. This is the ENCODE project. I'll skip that again. There's a study of pseudogenes. So you can see in the name pseudogene, even in the name itself, it sounds like this was a gene that once was and no longer is. It used to be a gene. It's broken. Well, the ENCODE project studied basically every single letter in the human DNA sequence. And since there's pseudogenes in the human DNA sequence, it's no surprise these were studied as well. Well, similar experiments have been done in other species, including worms and fruit flies. And the pseudogenes in these species have also been investigated biochemically for function. And not surprisingly, the number of human pseudogenes that have at least preliminary biochemical evidence for function mirrors what you see across the entire genome, about 80%. And similar numbers, 70-ish% in worms and flies, also hold true. Again, this is not genetic knockout experiments. It's not the gold standard. But it does set a trajectory. I don't think the evolutionists anticipated this sort of thing.
The fact